Good afternoon, everyone. We are live from the Teagasc Food Research Centre here in Moore Park in Ireland. I would like to introduce the other speakers. That's Claire Bourlieu and Christine Heerup. And I'm also joined by Myriam Grundy, who is one of the work group leaders of today's subject. And there's also Myriam Egan from our own research centre here in Moore Park, and she's doing the magic of the Zoom webinar in the background. So this is the fourth webinar in a whole series of webinars, the fourth webinar on food digestion. It has been recorded. So afterwards we will put it onto a YouTube channel, and I'll give you the details later. So today's topic is lipid digestion. I just want to share my screen with you just to get it into the context. Here we go. So Infogest is an international network, and the chair is Didier Dupont from INRAE in France, and the vice chair is Alain Mackie. And over the last couple of months, we dealt with a different working group. So we have 6 working groups altogether. So we had a webinar on in vitro digestion, on food, on absorption models. And today the topic is digestive enzymes, lipase, and lipid digestion. And the next 2 webinars here are here on the right-hand side, amylase and starch, and in silico models. So I hand over to Myriam.
Hello, everybody. So just before we start, I will present a bit of working group. So I'm just sharing my screen. So this is our working group, so share with Frédéric Carrière and myself. So we've got currently 40 members from 13 different countries. And just to present you briefly our objective. So the first one was to obtain a reliable source of lipase for the gastric phase. We wanted as well to optimize the activity measurement of the gastric and pancreatic lipases. We wanted to find an inhibitor as well for the different phases of digestion. And based on those results, we will eventually update the Infogest in vitro digestion protocol. And in the future as well, we would like to include other type of lipase with different substrate, activity on different substrates. So far we finish our first round trial where we measure the activity of the gastric and pancreatic lipase. And at the moment, we're having the second round trial going on, and we're performing a static digestion using the Infogest protocol. And we're doing those digestion on infant formula. So 10 labs are doing that at the moment. So if you like to be involved, or if you want any further information, feel free to contact either myself or Frédéric. So now, before we start, I'd like to remind you that you can ask a question using the Q&R button at the bottom of your screen. And it would be better if you could as well ask those questions during the webinar. Like the webinar is recorded, the presentation are recorded, so it will be easier for us if you do that as we go along. So Claire and Christine could therefore reply to those questions at the end of the presentation. So now I'd like to introduce Claire, who is a research scientist at INRAE in Montpellier now. And she's been working on the physico-chemical property of lipids, looking at the bioaccessibility and digestibility in particular in emulsion, and she's looking at the structure-function relationship. So the floor is yours now. So Claire, I think your presentation should arrive.
Hello to you all. So I'm really pleased to give this overview about the mechanism of lipid digestion in vitro and in vivo. And it reminds me of my first oral presentation for the cost Infogest in Le Croisic in 2011, and it's really nice to see that we have done a nice piece of a road together. So the outline of the presentation will be as follows. I will start with a short introduction, and then we'll detail what are the main stages of lipid digestion, their evolution with age. I will then point out the key lipolytic enzymes that contribute to lipid digestion before trying to describe the specificity of gastric and intestinal phase, the two main lipolytic phase of human digestion. I will finish with a wrap-up and a few take-home messages. So to start with, human digestive system is extremely effective in digesting lipids, and the coefficient of fat absorption reported in the healthy adults are above 95%. Lipids must convey roughly 40% of the total energy intake in the adults. They convey essential fatty acid, precursor of PUFA and eicosanoids, but they also help to solubilize liposoluble vitamins, including vitamin A and D, which are often deficient in humans. So the most part of the lipids in the diet come under the form of triacylglycerol. Around 80 grams per day are consumed, and a minor part under the form of polar lipids that are amphiphile molecules, phospholipids, glycolipids, and sphingolipids, representing 2 to 8 grams per day. Of course, the part of lipid in the diet evolves with age. It's really high during the neonatal period and during infancy to support the growth and development of the newborn and then of the infant. Lipid and lipid assembly will alternatively get constructed, deconstructed to vectorize energy or bioactive molecules. And probably most of the story is about how to cope with hydrophobic molecules in an aqueous environment. So it is possible thanks to the action of hydrolytic enzymes, they are called digestive lipase, that are very sensitive to interfacial properties at all water interface, and I will try to illustrate this on gastric lipase and in comparison on pancreatic lipase. And all these lipolysis interfacial reaction are in the forefront of more systemic effects that I will describe also in the presentation. It will be mainly focused on the digestion of emulsions, since it's my main object of characterization, and on the luminal part of the lipid digestion. So, lipid digestion, what are the stages? First of all, there is no digestion in the mouth or in the oral sphere, and the first stage of the digestion in humans is gastric phase. That will contribute to 10 to 25% of the total lipolysis of fatty acid present esterified on a glycerol backbone. The second stage will be the intestinal phase that will contribute, thanks to the secretion of pancreatic of the pancreas, to 56% of the lipolysis. Then the products of the lipolysis, free fatty acid and monoacylglycerol, will get solubilized and micellularized to be uptaken by the enterocyte, so the intestinal cell that will absorb this product and will re-esterify this product so that they can get assembled under the form of lipoprotein particles that are called chylomicrons after a meal, and that are the way to transport lipid assemblies through the lymph toward the target organs, liver, muscle, and adipose tissue mainly, but not only. And in this tissue will occur balance between storage and beta oxidation to release energy. Of course, as long as the lipid lipolysis products are released, they can trigger the release of satiety hormones, the main one being cholecystokinin, which is also represented on this graph, that will, by a feedback control, trigger the release of pancreatic secretion, but also a slowing down of gastric emptying. So now, does this lipid digestion evolve with age? The answer is yes. At birth, we have a mature human gastric lipase, but limited acidifying capacity. And we have an immature pancreatic lipase, but compensated partially by pancreatin-related lipase of type 2, for instance, or by salt-stimulated lipase. We also have limited biliary salt secretion. How does it evolve when we get older? Well, you can see on the gastric lipase activity reported here in the pharyngeal mucosa that the activity decreases again with age. And globally, as you can see in the synthesis that we have done for an article in Current Opinion in Clinical Nutrition and Metabolism Care, that the decrease in gastric lipase activity goes along with gastric motor function decrease and ACE delaying effect of lipid and gastric emptying, and also a decrease in pancreatic secretion. So within the COST Infogest, we contributed to this synthesis about physiological condition of lipid digestion and to the definition of some static adult model or some static infant model, and also to semi-dynamic or fully dynamic infant model that could try to mimic the digestion at a different physiological stage, and that I used during my research in complement with animal and clinical trials. So what are the key lipolytic enzymes at work for the digestion of triglycerides? So the two main ones are the gastric lipase, the human gastric lipase, and the pancreatic lipase with its cofactor, the colipase, at a molar ratio of 1:1 at least, that will hydrolyze triglycerides, but also triglycerides. In the pancreatic secretion, we will also find other pancreatid-related lipases, the carboxylester hydrolase, also named bisite-stimulated lipase, that will target and hydrolyze cholesterol ester, vitamin ester, phospholipids, and galactolipids. The phospholipase A2 that will hydrolyze phospholipids, and the pancreatic-related lipase 2 that will hydrolyze phospholipid, galactolipids, and monoglycerides mainly. In addition to these lipolytic enzymes, another discrete player is the alkaline sphingomyelinase secreted in the intestine to digest sphingomyelin into ceramides. So all these enzymes are amphiphilic proteins that will be affected by the ionic strength of their environment, but also by the pH, as you can see on this graph. And we can see that they are nicely adapted to the pH at which they must hydrolyze their substrate. And that's an important feature also of this pancreatic and gastric lipase is a very high specific activity of more than a few thousand units by milligram, which is used compared to classical microbial enzymes. This specific activity is, however, reduced in meal condition, which is not as optimal as the assay during which we determine specific activity. The secretions are well known. They are illustrated here, and you can find them also in synthesis in the literature for most of them, and have been adjusted in the COST Infogest digestive in vitro system for gastric lipase and pancreatic lipase. Now, if I go into the specificity of the two main luminal phase of the digestion, the gastric phase first, we can say that it is dominated by the action of human gastric lipase that is stereoselective, and thus will trigger the selective release of free fatty acid from the SN3 position of triacylglycerol, and it shares structural and functional homology with close mammalian analogs, including rabbit gastric lipase and dog gastric lipase that we have been using in our in vitro assay also. So if we look at the expression of this SN3 stereoselectivity of human gastric lipase, for instance, in vivo trial on human milk, we can see that facing the specific ratio distribution of short to medium chain fatty acid in the substrate, it will release selectively the substrate. And what is interesting is that those short to medium free fatty acid can be directly absorbed by the hepatic portal vein system, which are represented here on the right, and then delivered to the liver and beta oxidized there to generate energy. They are not able to acylate an hormone called the ghrelin, which is an neuroxygenic hormone, as it was shown by Lemarie et al. The gastric phase initiates a lipolysis by human gastric lipase, but it doesn't go very far because products of lipolysis will inhibit the action of the gastric lipase. Human gastric lipase is extremophilic and highly tensioactive. It has a high capacity of insertion in phospholipid interface, for instance, and no annex activity on phospholipid. We could also experiment that on a model infant formula having similar composition but different structure and space. So a coarse, minimally processed emulsion in comparison to two homogenized emulsion. And we could see that the kinetics of release of free fatty acid was really dependent on the surface available for absorption, and at equal surface with or without protein was not affected by the presence of this protein. To go further in the investigation of this capacity of penetration of gastric lipase, we used dog gastric analog of human gastric lipase, and could check its insertion in the liquid expanded zone of membrane, natural membrane, either from bovine milk or from human milk, as you can see here. And the lipase was able to get inserted quickly in massive amounts, and even more homogeneously in the human milk membrane, where long-chain PUFA would enhance the compressibility. So in terms of other specificity, which is really a key point, some drastic variation of pH occur during gastric phase, and will modify the colloidal behavior of emulsion that goes through the gastric phase. And this behavior is really determinant in the kinetics of lipolysis. There is also a synergy and interplay between lipolysis and proteolysis during gastric phase, and we could, for instance, evidence in vitro enhancement of adsorbed casein proteolysis and in vivo modulation of whey protein proteolysis, but with important variation linked with in vivo results. So if you want to illustrate further this gastric colloidal behavior in vitro and in vivo, you can see it on two milk emulsion. We can see that it's really dominated by lipid droplet aggregation and coalescence, and that aggregation of proteins occur around the lipid cluster. This is in agreement with the result obtained also by Gali et al. On oil body, for instance, almond oil body, in which gastric phase was dominated by aggregation and coalescence. Some magnetic resonance imaging, also data in vivo, report for fine emulsion a good stability in gastric phase, and for coarse emulsion a creaming and a destabilization with rapid emptying of the watery phase first and slow emptying of the fat layer afterwards. In vivo, in humans, still in the preterm, we could also experience modulation of gastric emptying and difference of lipolysis between fine emulsion based on homogenized human milks and the coarse emulsion based on non-homogenized pasteurized human milk. The delay in gastric emptying was explained by hormonal feedback triggered by higher lipolysis level, but also by a difference in colloidal behavior. So after the gastric phase, the intestinal redispersion of the emulsion is also a key factor. And in vivo, Armand et al. Reported indeed very early that fine emulsion or coarse emulsion could trigger distinct lipolysis kinetics and distinct lipemia with sharper peak for the fine emulsion. More recently, Borst and co-author introduced the concept of slow or fast lipid, which is also in agreement with this hypothesis that the kinetics of lipolysis between a fine and a coarse emulsion, for instance, or a fine emulsion and a non-spread fat will differ and will determine different lipemia postprandially and different metabolic fates. So a fine emulsion will trigger higher beta oxidation of ingested fatty acid and higher spillover of or loss of free fatty acids also. And this trend is an ACE in obese individuals. Similarly, Golding and co-author reported in vitro and in vivo difference of lipolysis kinetics for different emulsion having variation in the redispersion after the gastric phase in duodenal zone that would trigger postprandially different peaks of triglyceride appearance in the lymph. So now if we focus on the intestinal phase, we can say that human pancreatic lipase is regioselective of the position 1 and 3, and typoselective. It will have a faster effect on short chain and medium chain fatty acid and slower activity on long chain PUPA. It is more sensitive to interfacial quality than human gastric lipase that could get inserted in a variety of surfaces, and has a necessity to be with its cofactor, the colipase, to get anchored in interface in the presence of biliary cells. The adjustment of quantity of human pancreatic lipase and pancreatic secretion are regulated by hormonal feedback, including cholecystokinin secretion, and its action is completed by the action of pancreatid-related lipase, PLA2 acting on phospholipid, and PLRP2, for instance, acting on glycolipid, mono and diacyl diglyceride, for instance. Mono and D-galactosyl diglyceride, for instance. Sorry for that. So the action of human pancreatic lipase is also highly dependent on the amount of biliary salts present to solubilize the product of a human pancreatic lipolysis. And biliary salt secretion will get higher after a meal, reaching levels of around 15 micromolar, so well above the critical micellar concentration that help them solubilizing lipolysis products. So in terms of in vitro and in vivo correlation, using the cost Infogest static model, we could compare the lipolysis behavior and triggered lipemia obtained using two similar emulsions but with different interface, either dominated by milk polar lipids or by soy lecithin. And we could show, that it induced different lipolytic kinetics in the pancreas, in the intestinal phase, for instance, and that this difference in lipolysis would then trigger difference in lipemia in mice model. Similar results were obtained using again, submicronic, this time micronic emulsion of casein stabilized or soya lecithin stabilized flaxseed oil emulsion by Quedlow et al. With a good correlation between kinetics of lipolysis in vitro and in vivo in the rats. So what about going towards complete meal digestion? Not a lot of results have been reported in that direction, but that's probably an interesting direction. And we can see that for solid milk, generally, the lipolysis in gastric phase as well as in intestinal phase is much reduced. So in terms of take-home messages, we can say that the human digestive system is designed to efficiently digest free acetylglycerol, that the size of droplets in emulsion and their colloidal behavior in gastric phase, but also their dispersion in duodenal phase are determinant. They influence the kinetic of lipolysis and are then closely linked to a postprandial lipemia and metabolic fate of the lipid in general. The quality of interface also impacts, but to a lesser extent, unless a human pancreatic lipase inhibitor are present. And this quality of interface is more important for pancreatic lipase than for gastric lipase. The concept of slow or rapid fat is now admitted, introduced by Vorst and collaborators, and we still to gather knowledge about hormonal rate of control and associated dynamic adjustments to try to mimic it possibly in vitro or in silico, and also on the impact of intestinal lipid residues, lipids that resist digestion on intestinal microbiota or on other targets. Thanks a lot for your kind attention, and I'll be very pleased to answer your questions.
Okay, perfect. Thank you very much, Claire, for this really interesting talk. So we've got a few minutes to take some of the questions that we received. So if I start with the first one, absorbed lipids account for 95% of, or absorbed lipids equal 95%, but the sum of the gastric and intestinal lipid digested account between 65 and 80%. So what is the remaining part?
Yeah, so what I defined at the beginning of the talk is that usually healthy human would not lose more than 5% of lipid in the feces, so that would be the resultant. And when we are above 7%, we can define that we have a beginning of excessive loss or steatorrhea. When we talk about 10 to 25% in gastric phase and 56% well, in average in intestinal phase, upper part of intestine, we mentioned the acyl chain that are released. But when we are up to 66% of released acyl chain in the triacylglycerides, it means that we have only free fatty acid and monoacylglyceride. And this monoacylglyceride can be directly absorbed in the micelle by the enterocytes. So that's really the gap. The gap would be the possibility that we have to absorb monoacylglycerol, I think, in that question. And that's why we don't need to go to 100 or 96% lipolysis to get only 4% release in the feces.
Okay, thank you for your answer. So there was another question regarding pH and gastric lipase. So it is said to adjust the pH to 5.5 for the rabbit gastric extract. If gastric lipase reach its optimum at the pH, would it be effective when adjusting the pH to 3 in the gastric digestion?
So I think that you've seen in my slide the example of the evolution of specific activity of human gastric lipase with pH, and that it's a curve with an optimum, but then that decrease but do not drop. And if you look at the remaining activity at pH 3, it will be different between a human and a rabbit gastric lipase, but you still have got some residual activity. And as I point out, that the initial activity was extremely high. So for human, more than 1,000 units by milligram, the remaining activity will be far enough to hydrolyze 10% of the acid chain that are present. But so please go and look at the specific activity against pH curve and you can also be reassured on this residual activity at pH 3.
So I think we've got time for one last question. So as we answered it already, but I just read it out loud. I guess that the current Infogest protocol does not include phospholipase. Does it mean that it is not suitable to study in vitro digestion of lipid for food rich in phospholipid?
I think that we mentioned also in the first version of the protocol that we could adjust and go for a mix of pancreatic lipase and PLA2 lipase when we had to digest specifically emulsion containing a phospholipid. So in one of the example I gave in Leconte work, we did that. So we complemented the Infogest protocol with some phospholipase A2 because it was food rich in phospholipid. But you will still find some annex activity in pancreatin source as well. But maybe Myriam, you have also comment on that answer.
So like I indicate in my answer, is something really we would like to do as part of the working group for, because we don't know, and it probably vary from batch to batch of pancreatin, but it's something that would be really interesting to try to determine actually how much phospholipase activities there is in the pancreatin.
I might actually come in there as well, because at the time of writing of the Infogest protocol, there were very few people that used it. So there was no scientific consensus, and that's why it was not included in the protocol. As Myriam said, you know, there's a drive to look into it a bit further, and if there is a consensus, by all means, it will be included in the future. So that's something to look out for. That's not the only part. You know, there might be other things to be included in the future. Okay, so I might take over. Claire, thank you again. It was an excellent talk, as usual.
Thanks a lot for the invitation.
Very good. So as Myriam said, we try to answer all the questions live, and any questions that haven't been answered live, we can answer afterwards. So type it in, keep typing, and we look after it. So at the moment, we are 160 people, live attendance from all over the world, which I'm really delighted about. Also a big hello to those that cannot attend live and watch us on YouTube afterwards. So mainly the people from overseas. So the next talk is by Christine Heerup. As I said earlier, we usually have two speakers. The first speaker is generally a senior speaker giving an overview of the subject. And it was our wish to include a second speaker, such as a PhD student. So at the moment, there are not many conferences where PhD students can present their results. So I'm actually delighted to introduce Christine Heerup. That's our first PhD student in this webinar. Christine is a PhD student in the University of Copenhagen, Department of Pharmacy, and she's a participant in the infant brain project with primary focus on in vitro pediatric lipid digestion of bioinspired emulsion. So I hand over to Christine. Here we go.
Hi, my name is Christine Heerup, and I have the privilege of giving today's talk on in vitro pediatric gastrointestinal digestion of bioinspired oil in water emulsions with recombinant human gastric lipase and pancreatin. This work is part of the research that I've done during my time as a PhD fellow at the University of Copenhagen, and it's part of the Infant Brain Project. Before I start, I would just like to acknowledge all the people who helped me do this research. I could not have done it without you, so thank you very much for that. I would also like to thank our funding partners. First, a little bit on the lipid digestion in infants and the role of the milk fat globule membrane. During the first 6 months of life, infants rely on mother's milk and infant formula as their primary source of nutrition. Lipids constitute approximately 50% of the daily energy intake and are vital for the growth and development of the infants. Especially the long-chain polyunsaturated fatty acids, the LC-PUFAs, are needed for optimal brain development. Lipid digestion in infants initiates in the stomach by gastric lipase, which is responsible for 10 to 30% of total lipid digestion. The remainder is digested in the small intestine by the pancreatic triglyceride lipase, the bile salt stimulated lipase, and the pancreatic lipase-related protein 2. However, the structural organization of lipids in mother's milk is very different from infant formula. In mother's milk, the lipids exist in milk fat globules, which are covered in a complex tri-layered membrane structure called the milk fat globule membrane, or MFGM for short. And this is not found in infant formula in that way. This MFGM plays an important role in lipid digestion in infants, since the lipases in the infant's gut has to interact with the MFGM in order to get access to the lipids inside the milk fat globules. During the last 100 years, give or take, a lot of studies have been conducted which indicates a correlation between breastfeeding and cognitive development. A meta-analysis conducted by Anderson et al. In 1999, which is shown here on the left, showed that the IQ in breastfed infants was higher than in formula-fed infants, and that longer duration of breastfeeding also had a positive effect. They also showed that the difference was bigger in low birth weight infants compared to normal or mixed birth weight infants. The difference between having been breastfed or formula-fed as an infant could even be measured in adults in terms of adult IQ, educational attainment, and income, as shown by 2 other studies. So, how to improve the cognitive development in formula-fed infants? Well, in a Swedish study, the Tumor study, they developed a new infant formula enriched with bovine MFGM, which they tested against a standard infant formula and breastfeeding. They found that at 12 months, there was no difference in the cognitive development of breastfed infants and the infants fed the experimental formula containing the MFGM. The infants fed the standard infant formula, however, still showed significantly lower cognitive development compared to the breastfed infants. This leads up to the project that I will be talking about today, which is the Infant Brain Project. In Infant Brain, we aim to develop new infant formulas to promote brain development. Aside from milk fat globules, milk also contains extracellular vesicles. In Infant Brain, we use emulsifiers derived from bovine dairy process streams containing milk fat globule membrane as well as membrane from extracellular vesicles to make bioinspired lipid droplets for infant formulas that are more similar to the milk fat globules in mother's milk and are more easily digested. The idea is that the optimized lipid digestibility results in improved bioavailability of LC-PUFAs such as DHA, which, as I said before, has been found to be important for optimal cognitive development. To study the digestibility of the bioinspired emulsions, we developed a digestion model simulating lipid digestion in infants. In this model, the digestion is quantified 2 ways. Indirectly, by continuous titration of the released fatty acids, and secondly, to validate the titration method, samples were taken during the in vitro digestion for chromatographic quantification on HPLC ELSD. This digestion model consists of first a gastric step, followed by an intestinal step, and lastly by a back titration to pH 9 to account for any free fatty acids that were not titrated during the gastrointestinal steps. Most previous in vitro digestions have been carried out using microbial lipase or animal gastric lipase as a surrogate for human gastric lipase. In this digestion model, we use recombinant human gastric lipase. This recombinant human gastric lipase is produced by Bioneer in mammalian cells, and a study by Cezanne has found that the gastric lipase surrogate that mimics the lipolytic activity of the human gastric lipase the best is in fact the recombinant human gastric lipase. For the intestinal step, we used a porcine pancreatin. For the development of this digestion model, we used 2 different substrates to show the versatility of the model. One was a ready-to-drink infant formula, NAN1, And the other was an in-lab prepared 3.5% simple oil and water emulsion stabilized with pure soy phosphatidyl-choline. And that's emulsion is called the SPC emulsion. We did some cryo-SEM imaging to see how they looked, and the cryo-SEM imaging showed that the surface of the NAN1 lipid droplets was quite rough and had a lot of protein aggregate attached to the surface. On the other hand, the SPC emulsion had lipid droplets with a very smooth surface with what appeared to be small lipid droplets of vesicles attached to the surface. We also determined the droplet size distribution and found that the droplets in NAN1 was generally smaller than the droplets in the SPC emulsion. So here are the results from the in vitro digestion of the two substrates. And what we have is 15 minutes of in vitro gastric digestion with 3.75 TBU per ml of recombinant human gastric lipase, followed by 90 minutes of intestinal digestion with 26.5 TBU per ml of pancreatin. The digestion of NAN1 and SPC emulsion resulted in two sets of data. One is the continuous titration of free fatty acids, shown on the left y-axis, and the other is the digestion products, the free fatty acids and monoacyl glycerols, measured by HPLC at time 0, after the gastric step and after the intestinal step, and is shown on the right y-axis. As you can see, we found really good correlation between the titration results and the HPLC results. We therefore concluded that quantification of lipid digestion by titration is an accurate method for quantifying lipid digestion and can be used independently. With a digestion model in place, we moved on to look at the digestibility of the bioinspired emulsions. As I mentioned previously, milk contains both milk fat globules and extracellular vesicles, and we wanted to investigate the potential of using bovine MFDM and EVM fragments as emulsifiers. We therefore made bioinspired emulsions with different bovine dairy process streams, where two contained mostly MFDM fragments, but also some EVM fragments, namely a whey protein concentrate enriched with phospholipid and a butter serum enriched with phospholipid. And the other two process streams were filtered to contain primarily EVM fragments and were a whey protein acid extract enriched with EV material and a micellar casein isolate enriched with phospholipid. In addition, we used two referent emulsifiers, namely a protein reference consisting of whey protein concentrate and a phospholipid reference consisting of pure soy phosphatidyl-choline. These references were chosen since whey protein concentrate and soy phospholipid in the form of soy lecithin are found in most infant formulas. We also did a compositional analysis, and that revealed that, as expected, we found very different amounts of phospholipid in the two reference emulsifiers, and also different protein content, exactly as expected. With regards to the 4 bovine-derived emulsifiers, we found different total amounts of phospholipids, but the composition of the phospholipid subspecies was quite similar between the 4 emulsifiers. With regards to the total protein content, we found the same protein content in 3 out of the 4 emulsifiers, but slightly less in BSPL. In contrast with the phospholipid composition, we actually found that the composition of the subspecies of protein was very different between the 4 emulsifiers. Here we have the results from the in vitro digestion of the bioinspired emulsions, and I've shown it as a summary of the digestibility, just to make it a bit more simple, where the solid part in the bottom is the gastric digestion, the striped part in the middle is the intestinal digestion, and the checkered part in the top is the back titration. Together, the height of each bar represents the total digestion of each emulsion. For this study, we lowered the duration of the intestinal digestion step to 25 minutes compared to the 90 minutes used during the development of the model to avoid coalescence of the lipids in the digest. What we saw is that of the 6 emulsions, PL had the lowest digestibility of them all, which is interesting considering that phospholipids are found in infant formula. Also interesting is that whey protein concentrate, which contains no phospholipid, had the 2nd highest total digestion. Surprisingly, the digestibility of WPCPL and BSPL, the blue and green one, was significantly different, even though both were made with emulsifiers containing MFGM fragments. And the same was observed for WPC-AEV and MCI-PL, the red one and the pink one, both of which were made with emulsifiers containing EVM fragments. But in general, the highest digestibility was observed in WPC-AEV, which is the red one. To investigate what drove the digestibility of the emulsions, we looked into the emulsifier content, and we found that while the neutral fat and phospholipid content was not correlated with total digestion, a significant correlation was observed between the protein content and the emulsion digestibility. But due to the low amount of emulsifiers tested, the amount of information gained from these in vitro digestions was limited. In an attempt to make more data points, we used the same emulsifiers as before and substituted 50% of the emulsifier whey with whey protein concentrate to increase the protein content, and 50% with soy phospholipid to increase the phospholipid content. Thereby, we got a lot more data points for use in our statistical analysis. Since the digestibility of the PL emulsion was so low compared to the other emulsions, it was not surprising that 50% substitution with soy phospholipid in general decreased the digestibility of the emulsions. However, we can also look at it differently. We can say that adding MFGM or EVM material to a pure soy PL emulsion actually improves the digestibility of that emulsion. And what was surprising was actually the effect of substituting 50% of the emulsifiers with whey protein concentrate. We found that in both WPC-PL, the blue one, BSPL, which is the green one, and MCI-PL, the pink one, the substitution with whey protein concentrate increased the digestibility, and in 2 of them, WPC-PL and MCI-PL, the blue and the pink, the resulting mixed emulsions had even higher digestibility than when both of the emulsifiers were used alone. In WPC-AEV, however, the substitution with whey protein concentrate did not improve the digestibility. These results indicate that emulsifying the lipids in infant formula with emulsifiers containing MFGM and EVM materials shows great promise. Since PL and WPC is used in most infant formulas, using MFGM and EVM derived emulsifiers in infant formula has the potential to improve the digestibility further due to the positive combinatory effect that we've observed. To get an idea of which components of bovine milk has the greatest effect on the digestibility, we conducted a series of correlation analysis once more on the larger dataset. Taking into account that the phospholipids used in this study was not bovine derived, but of soy origin, and that higher levels of soy phospholipids did not improve the digestibility, we did not include any of the emulsions containing the soy phospholipids. In other words, we only looked at emulsions made solely with emulsifiers of bovine milk origin. What we found was that there was no correlation between the total digestion of the emulsions and the total emulsifier content of phospholipid and protein. However, we did find a positive correlation between the digestibility and the content of the protein beta-lactoglobulin, and a negative correlation between the digestibility and the content of casein. We also investigated the subspecies of phospholipids and found no correlation. These results indicate that the specific protein composition has great influence on the digestibility. They also indicate that emulsions made with bovine dairy-derived emulsifiers containing a combination of high beta-lactoglobulin and low casein content likely will show high lipid digestibility. So the take-home message. In vitro pediatric gastrointestinal digestion model was developed with recombinant human gastric lipase and pancreatin. We investigated the in vitro digestibility in bioinspired emulsions and found that MFGM and EVM ingredients have different composition, and used as emulsifiers, they give different digestibility. Higher digestibility in bovine milk-derived emulsifiers were found compared to using soy phospholipid as an emulsifier. We also found that 50% substitution with pure soy phospholipid decreased the digestibility, and that 50% substitution with whey protein concentrate improves the digestibility, and it was attributed to high levels of beta-lactoglobulin and low levels of casein. Using MFGM and EVM-derived emulsifiers in infant formula to improve the lipid digestibility therefore shows great promise and is definitely worth looking more into. And with that, there's nothing else to say, then thank you for your attention.
Thank you, Christine. It was a very clear presentation. So we have a number of questions, quite a lot of questions, but we only have time for probably 2 questions. So I start with the first question. Did the bioinspired milk fat global membrane have phospholipid tri-layer membrane structure? If yes, could you please mention the reference paper?
Okay, so when we did these emulsions, we did it on MFGM material. So in the bioinspired emulsions, we did not end up with a tri-layer membrane structure as such. In a previous article, they presented the Nuturis phospholipid droplet. Gallia presented an article on that. And they have shown from microscopy that the structures of these bioinspired emulsions, which we are also doing, they have more similarity to the human milk droplets, but they will not have the complete tri-layered structure. This tri-layered structure will be disrupted during the dairy processing, but the surface in overall will mimic it more than if you simply use soy phospholipid or whey proteins to cover the surface.
Very good, very good. Of course, one of the obvious questions that the audience would like to ask is the recombinant human gastric lipase, is great. Is it commercially available, or was it within the project?
Yes, so this project is done in collaboration with Bioneer, but anyone can contact Bioneer to order the recombinant human gastric lipase, and they just have to contact Annette Mulets, who is my supervisor, and I will happily give out the contact information for anyone who wishes to use the recombinant human gastric lipase, so it can easily be bought and used for other studies.
Excellent. And I have one last question, and I was interested in the fact that you used EV-enriched infant formula. So there's a question from Charlotte Oliver. How different are EVs from whey protein concentrate to milk EVs?
Yeah, so the EVs, the difference lies in the difference in how they're produced. So the EVs will only contain the membrane layers arriving from the endoplasmatic reticulum, whereas the milk fat globules will have a membrane that contains first the double-layered membrane or the single-layered membrane from the endoplasmatic reticulum, and then the apical layer. I just mixed it up, sorry. The tri-layered membrane comes from first a single-layered membrane from the endoplasmatic reticulum, covered by a double-layered membrane from the apical cell membrane in the breast tissue. Whereas the EVs, they will only contain the cell membrane double-layer structure. So they lack the inner membrane structure from the endoplasmatic reticulum. And that's the primary difference. And of course, they do not contain any lipids They just contain the water resolvent of the cell.
Okay, very good. Thank you, Christine. Well done, well answered question. So I would like to thank both speakers, Christine and Claire, and also Myriam for moderating. So I have four more slides before we finish on the hour. So give me a second. I just share my screen. Okay, as I mentioned earlier, we have 6 working groups. So today we are on working group 4. The next 2 are working group 5 and 6. And I might mention that if you have a story that you want to share with us here on the webinar, write to the work group leaders of working group 5 and 6. I may also mention that so far we didn't get too many offers to talk. Okay, so it is up to the working group leaders to decide who speak or who don't. So it's not me. So we do have one speaker for the next webinar on the 6th of January in 2021 already. So that's the starch digestion amylase. And we have one speaker so far, Daniela Freitas, who's based here in Moor Park, but she will present some of her work in INRAE. The second speaker is to be confirmed. So again, if you want to be a second speaker, please write to the working group leader or write to myself. The next one after that is on the 3rd of February. So every first Wednesday of each month at 2 o'clock Dublin time are the webinars. So the next one is in silico digestion models, and the speaker will be confirmed next week. If you would like to join the network, please write an email to Nathalie or Didier Dupont. Also, you can follow us on the LinkedIn Infogest group or myself. If you want to post something on LinkedIn, just type @Infogest. So if you're looking for a person or if you have a paper that would like to share, just write @Infogest. We will put this webinar up on our YouTube channel. If you scan that on the top right-hand corner, just Google Infogest and webinar. And here you see already the first webinar, second, third, viewed a number of times here, several hundred times already, which is great. Okay, so that's my last slide. I see you all on the 6th of January. Again, thanks to the presenters today and to Myriam. Goodbye from Ireland and Sloan.